Go ahead.
Okay, thank you, Rachel, for organizing this seminar. I hope everybody is well, everybody's healthy, everybody is safe given the circumstances. And so, I mean, what I'll do, I'll start with an introduction, and then I'll tell about some of the projects that we're doing in the lab at the moment. I mean, this is my usual slide when I cover slide, but of course, now we need to change it to Institute of Life course and medical sciences. And if you look at terms of life course, I mean, the human life course is, you know, from conception, birth to middle age, and then to old age. And of course, my my research in particular focuses on old age, on the elderly individuals, which, as you know, is a very timely topic. The incidence of age-related diseases is increasing, and is very timely as well, given what's happening with the pandemic. So you might have seen slides like this before, but this is the COVID-19 mortality rate per age. This is data from China, from about from the first pandemic, the first peak that they had there. And as you can see, mortality below the age of 50 is very low. At the time in China, nobody under the age of 10 had died. So it was actually zero in young children mortality. I think there's been a few children, unfortunately, who have died now. But of course, the point is that the mortality of COVID-19 is particularly high at, older ages, after age 70, 80. That's where it becomes a problem. So this really is of emphasis. Okay, yes, you have this virus, but the virus is primarily affecting older individuals. So there's something about the aging process and immunosenescence and aging of the immune system that predisposes people to this virus. So if we could somehow slow the process of aging, then this would have impact on not just diseases like cancer and heart disease and neurodegenerative diseases, but infectious diseases like COVID-19 and seasonal flu as well. Now, unfortunately, we don't know yet exactly why we age. We just don't understand the mechanisms that drive the process of aging, the molecular cellular processes. There are some hypotheses, and I'll mention them today, but we don't really understand why we age at the molecular mechanistic level. And I mean, this applies also to the immune system. So having said that, there's been a lot of progress in our understanding of longevity pathways, I would say. Because now we know that the process of aging is plastic. We can accelerate and retard aging via genetic manipulations and via dietary manipulations. So the first gene associated with aging, I'm sure many of you are aware, was H1, identified by Tom Johnson and colleagues. And this was the first demonstration that one genetic variant has a significant impact on the process of aging. And that was done in C. elegans. And now we know many other genes, as I'll come back in a minute. And we also know of dietary manipulations of aging, like dietary restriction, that in model organisms, in animal models, allows us to retard the process of aging. And I mean, this is something I've always found fascinating. And so back when I was a PhD student, I started this collection of databases called the Human Aging Genomic Resources, which they've grown. Now they're the benchmark databases in the field. They have hundreds of thousands of visitors, hundreds of citations. And I mean, I'll focus firstly on our GeneAge database of aging-related genes. And I'll point out as well that we have those interested in the latest biology of aging news. We have pages on Facebook and Twitter with the latest papers. But anyway, so going back to the GeneAge database, I started this as a PhD student because I was fascinated by this ability of individual genes. You tweak a single gene and you increase lifespan significantly. You retard the process of aging. I thought it was incredible. I mean, if we could do that even to a smaller degree in humans, that would be amazing. So I started collecting and curating all the data on these genes. And so now the latest version of GeneAge database, which was updated just a few months ago, actually, shows that really we've done huge progress in our understanding of the genetics of aging and longevity in model organisms. So now we know of over 2,000 genes in model organisms that extend lifespan. That affect the aging process. They could retard aging or can accelerate it. So it's quite remarkable. It's what I call the gerontome. And a lot of this is in worms, in C. elegans. So nearly 900 genes in C. elegans, including a single gene manipulation that extends lifespan by tenfold. Also, mice and flies. So in mice, we can extend lifespan by roughly up to 50% really by single gene manipulation, which again is remarkable. So this is really a demonstration of how much we've advanced in our understanding of genetics of aging in model organisms. And I mean, if you're interested, I mean, we published a paper a couple of years back, and obviously you're welcome to visit our database. And I mean, as we go along, I'll mention some of the people from our lab, in particular recent or current members that have been involved in the various projects. So in the particular project of the Human Aging Genomic Resources, Daniel and Emily are no longer in our lab. I mean, Emily is still a PhD student in our institute, but they previously worked on this. And Inez and Zoya are currently working on our databases as well. Although Zoya, at the moment, she has a different project. She has, just a week ago, she had this adorable baby girl, so she has other priorities other than our databases at the moment. Now, so one of the things I would point out about this advances, which although it's remarkable that we're knowing about all of this genes associated with aging, it's a lot of data. It's over 2,000 genes. So in a way, together with next generation sequencing approaches, which I'll come back to in a minute, really the bottleneck of research is now not just generating data, but it's actually analyzing data. So biology and biomedical research is becoming a mathematical, a statistical problem, in that really we need to have the analytical tools to interpret and analyze and process all of the data that we're generating. So that's something I've realized back when I was a PhD student, actually. So I did my PhD in cell senescence. So I was an experimental biologist, and I shifted to the happy life of computational biologist when I was when I realized this. So actually, that's a photo of me when I was a PhD student. And yes, I know that there's two things I would say about that photo. First of all, yes, I did think my hair was cool back then. And secondly, given the lockdown, give it a couple more months and probably I will look like that again in the future. But so I shifted when I was a PhD student from an experimentalist to a computational biologist. And though, I mean, I will say we still do some experiments now in our lab. I would say most of what we do is more on the computational side. So, as I'm sure many are aware, our lab is the Integrative Genomics of Aging group, and we do some experiments, but mostly computational approaches, including various computational bioinformatics, systems biology approaches, machine learning. We focus dietary manipulations of aging. We do some experiments, particularly in cells. As I said, I'm actually a cell biologist by training. And we do various comparative and evolutionary genomics, functional genomics, and so on. So I'll start by giving a very brief overview of what we do, and then I'll mention, without actually going into a lot of detail, to some of more recent projects. So I guess we've done a lot in terms of functional genomics and transcriptomics, from microarrays to RNA-seq. And I'll mention an example of RNA-seq in a few minutes. We've done a lot of whole genome sequencing. I think one of the biggest projects from our lab was the sequencing of the bowhead whorl genome, which we did in the CGR, in the Center for Genomics Research in Liverpool. I won't go into details, but that was quite a big project a few years back. We've done some genetic association analysis. So we've done a lot of genetics, genomics, and transcriptomics. That's something we're very experienced with. And as I said before, we do a lot of computational, from large-scale data analysis, data integration, visualization, modeling. I'll talk about network approaches in a little bit. We do some evolutionary analysis as well. I won't have time to go into, but we've done some work, for example, in looking at cancer-related genes in whales. We have a bioarchive preprint on that. And that's been the work, recent work of Daniela. She's now in Seattle, but she used to be in our lab. And we do a lot of methods development from epitranscriptome tools. This is particularly in collaboration with Jiameng at XGLU University in China. We also do some software development. This was the work particularly of Gianni, who was a PhD student in our lab until last year. And he did quite a lot of software development. For example, for the analysis of flow cytometry data, he developed an R package. We've done some, more recently, we've had a couple of projects and a couple of papers on also advancing cryopreservation techniques. Not something I'm going to go into detail today, but that's kind of a side interest that, so we published a paper just a couple of months back by Alex Murray, who was a master's student on work with Drosophila larvae, and also Soren, who was in our lab a few years back, also on cryopreservation of human cells. So we've done quite a bit in terms of that I won't go into detail, but that's something I'm still interested in. So I'll mention a few recent projects. I guess conceptually my interest is going back to what I said, that, okay, we're generating lots of data. We know of lots of genes that influence aging, that influence diseases and so on. But really, genes don't work individually. They work by interacting with each other and with the environment. So we have to understand the connections between the genes, the connections between the components in the cells. We have to take these systems approaches to understand complex biological processes, including aging and longevity and well, not all, but most age-related diseases. So trying to understand the whole from its parts, the so-called systems biology paradigm. And so I'll show you a couple of examples of this. Again, I won't go into a lot of methodological details because I want to mention various projects we've done. But most of what I'll mention has either been published or is now in preprints. So one of the things that we did a few years back that I thought was quite interesting was did this systematic analysis of the gerontome. That is, we looked at the GeneAge database and we applied various methods. One very simple, I won't go into details again, but one very simple method that we can employ to reduce the complexity of the data is to cluster or to group genes into common functions and processes. So that's something we've done. So we've actually classified genes in GeneAge as pro and anti-longevity, depending on their effects. So for example, an anti-longevity gene would be, say, the growth hormone receptor that you knock out in mice and it extends lifespan. So that's anti-longevity. And pro-longevity would be the opposite. But then we can group genes into various pathways. So according to the various model organisms that we have, we can identify the major pathways modulating longevity and aging. So, most of this would be expected. So for example, in mice, we see pro-longevity cell cycle regulation and p53 signaling. And in anti-longevity, we see as not surprisingly growth hormone, insulin signaling, IGF-1 signaling. In flies, we see as pro-longevity hypoxia activation, and as anti-longevity, again, IGF-1, PI3K, which also related to IGF-1 and growth hormone signaling, and oxidative phosphorylation. And in worms, we see as pro-longevity autophagy, and as anti-longevity mTOR signaling and oxidative phosphorylation. So pathways that by and large you would expect. What I think is interesting, however, is still that, okay, this is an unbiased approach. We didn't select these pathways. It's unbiased. It's a data-driven approach of looking at the major pathways that modulate longevity. So, we did our analysis in this paper that I won't go into detail. Actually, one of the things we did was look into correlations between genes associated with the process of aging and diseases, because that's one of the big questions. Okay, you have a process of aging and you have susceptibility to age-related diseases, but how do they interact, or how do they relate to each other? So let me focus on one example, which is the relationship between aging and cancer. I'm sure you're aware cancer in age-related diseases increases incidence exponentially with time, and there's various potential mechanisms for this. Again, I mean, I did a review, Nature review, a few years back, but there's various possibilities. We have, maybe there's an accumulation of senescent cells. There's cells that stop dividing, which is something I'll come back to as well in a little bit. Maybe there's an increase in inflammation with age that facilitates cancer development with age. You know, there's various possible mechanisms of fostering, but also preventing cancer development in old age. So what we've then done was we looked at the transcriptome between aging, cell senescence, and cancer. So we took publicly available data from GTX for human aging and TCGA for cancer, and then we derived lists of genes differentially expressed with age, in senescence, and in cancer. Now, of course, senescence is from cell lines, but aging and cancer is from different tissues. So we have data for various tissues of changes that occur during aging and changes that occur during tumor development or in cancer. And then we compare them. So we published this last year. I'll just summarize the results. So what we find is that there were a lot of tissue-specific patterns. So the broad message, I guess, was that changes in cancer and aging occurred in opposite directions. So you see genes going one way in cancer and the opposite way in aging in the same tissue. And these tended to be processes related to cell cycle. So you didn't have an increase in cell cycle promoters, genes, or genes that are associated with cell division in cancer, as you might expect, but you tend to see a decrease or a downregulation of those genes during aging, and also processes related to the immune system. So this was the general approach, but there were a couple of exceptions, like the thyroid and in particular the uterus. So the uterus seems to be quite the opposite, in that you're aging, you actually have an increase in gene expression similar to what you see in cancer, which I thought was quite interesting. How you exactly interpreted this results, I think, is up to discussion. I mean, my initial instinct was that this was a way of maybe, you know, aging processes almost fighting or trying to reduce cancer by downregulating genes associated with cell cycle. Having said that, I mean, you know, others, I mean, one of the reviewers from our paper, Jim DeGregory, actually said, well, I think it's the opposite. I think by having fewer cells that are dividing in a tissue, you create an environment that's more susceptible to cancer development, which I think theoretically makes sense as well. So how interpreted, I think it's not clear to me yet, and I think it would be interesting to explore what this all means. I mean, if you're interested in more details, as I said, the paper was published last year, and this was the work of Cassett, who's the first author in the paper. He's a PhD student in our lab. And well, a few others collaborated as well. Now, the other thing we looked into is cell senescence, as I mentioned before. So cell senescence is, you know, essentially, I tend to define cell senescence, a senescent cell, as a cell that should be dividing but isn't. So this was observed initially in vitro, human cells that divide but then stop, but it's been observed in vivo as well. So what we have is, we have a number, I mean, the hypothesis, I guess, is that with age, there's an accumulation of these senescent / non-dividing cells. And in turn, they secrete inflammatory cytokines, which are called SASPs, secretory-associated senescence phenotype, which in turn contributes to inflammation and tissue dysfunction. There is evidence. I mean, we published this review with Jean Passos in Newcastle. Well, he's now in Mayo Clinic, actually, in the US. But a couple of years back, there's a lot of evidence, I think, from mice that senescent cells are important for tissue degeneration of, well, at least some tissues. Whether it's important in human aging is a different matter, but certainly there is evidence that it's important in aging in mice. And so one of the things we've done is we've developed a database of genes associated with cell senescence. These are genes that when you genetically manipulate it in human cells, they either retard or accelerate the phenotype of cell senescence. So as I said, senescent cells are cells that stop dividing, and they tend to have this particular morphology. So you see here young fibroblasts, and these are senescent fibroblasts, who tend to have much more heterogeneous morphology. And that's a photo actually from my PhD. So we derived this 279 genes associated with cell senescence, and depending on how the phenotypic impact has, we can define genes as promoting or inhibiting cell senescence. So a gene, if you activate cell senescence, would be an activator of cell senescence, and vice versa. And then we've done various analyses. So one of the things we looked into was the cell senescence genes in normal human tissues using the GTX data. So we have inhibitors of cell senescence in various tissues, human tissues, and promoters of cell senescence. So there's not much of a signal regarding inhibitors of cell senescence, but in terms of promoters of cell senescence, there is an overrepresentation of tissues in which these genes are overexpressed. I mean, clearly it's not all the tissues. For example, in muscle, that doesn't seem to happen, and in other tissues. But there are more tissues than expected by chance with an overexpression of promoters of cell senescence, which I think is an interesting sign that, okay, you have an accumulation of senescent cells and non-dividing cells in human tissues with age. In some human tissues with age, actually, let me rephrase that. It's also interesting to see the uterus again as an exception. You actually see an overexpression of genes underexpressed in cell senescence in the uterus. So clearly it doesn't happen in all the tissues. It happens just in some of them, even though overall it seems the majority is an overexpression of genes. And then we did various networks. So I'll take a step back to focus a little bit on what network approaches are. So, I mean, there's a lot of different network approaches that we can employ. I mean, we've done this big review in Briefings in Bioinformatics in 2018 that by my former PhD student, Sipko van Dam, that I strongly recommend for a review of network and co-expression networks in particular, because there's lots of different approaches you can employ to study gene networks and protein networks. One of the things we've done is just what's called a guild by association. So it's fairly straightforward. So basically, if you have a network, so let's say these are network of proteins, and in red you have proteins associated with aging or longevity, let's say from the GeneAge database. And in blue you have proteins that have not been associated with longevity, not been studied yet in the context. But if a protein interacts with a lot of proteins associated with a phenotype, let's say longevity, then by chance it's more likely to be related to that phenotype. And of course you can do a lot of statistics behind it to derive a p-value and out of it and do a ranking of genes. So we can use this, what's called this guild by association approach, to identify candidates for various processes. So we did it for longevity, we've done it for diet-mediated life extension, which was something we published in PLOS Genetics a few years back, together with a number of collaborators. So we've shown that you can use these network-guided approaches to derive genes associated with particular phenotypes, including in the context of longevity and aging. The other thing we've done is we've developed actually this tool. This was developed by Sipko some years back when he was a PhD student in our lab. We developed this tool called GeneFriend. So basically, this is a co-expression map. So what we do is now we use RNA-seq data. So we have over 80,000 transcripts. So we get all the RNA-seq data we can get our hands on, and then we build a whole genome co-expression map. So we look for 80,000 genes by 80,000 genes. We look at how strongly the genes are co-expressed with each other across all of the existing data. And then we can use the guild by association methods to associate, to identify new genes with a given process. So if you have a number of genes associated with with aging or with osteoporosis, with any process or disease, you can then look at which, so you have a seed list of genes, you can then look at other genes associated with that seed list of genes. And you can also, if you have one gene that you don't know the function very well, you can also look at which genes are co-expressed with. So if a particular gene is co-expressed with mitochondrial genes, for example, then well, there's a probability, there's an hypothesis that it will be have a function in mitochondria. So I mean, I would say it works quite well for some genes, not so much for others, but I think generally speaking, it works quite well. So I would invite you to visit our website. And this project was started by SIPCO, but it's now being developed by Priyanka in our lab. So again, I would invite you to look at our GeneFriends tool. Now, one of the things actually we did initially when we developed GeneFriends was look at aging. We found some genes, but it wasn't, I mean, statistically, the results were not that brilliant. I think because aging, you know, there's lots of different tissues, there's lots of different processes. But then we tested trying to look for new genes associated with cancer. And we found this one gene, C1orf112, which was strongly co-expressed with cancer-related genes and often overexpressed in cancer. And then we looked into HeLa cells. We did which is silencing with siRNA. And we showed, which is what you see here, then when you silence C1orf112, it reduces cell proliferation in HeLa cells, so in the cancer cell line, compared to a negative control. So, well, that doesn't by itself prove it's cancer-related. It fits with our hypothesis, and it shows that at least the gene is doing some function. And so for the past few years, we've been quite interested in this gene. We actually created collaboration with Paul Potter in Harwell. We created some conditional knockout mice. So actually, I got in touch with Paul because I noticed that they had some knockout mice there, but well, they were trying to generate some knockout mice, but they were not viable. So what you see here, this is a normal mouse embryo, and this is a blob of a knockout from C1orf112, whose mouse homologue is this BC055324. So again, it shows that, you know, this gene is doing something. Then we created this conditional knockout mice without going into a lot of detail. After about a year of, so this is a conditional knockout in which you give the mice tamoxifen, and that ablates the gene in most cells. But we didn't notice any major phenotypes. There was, I think the only phenotype that the animal facility reported was they had very small testes, these animals. Other than that, there was no major phenotype. But it is something we're still, I mean, we don't have the mice anymore, we ran out of funds, but we still have frozen embryos and constructs. So, well, certainly if we had the funds, it would be quite interesting to explore this further, maybe cross these animals with a cancer model. So now we're more focusing on cell studies of C1orf112. And this is mostly the work of Jacob, who, I mean, we have a CRISPR knockout cell line that allows us to study this this gene in vitro, which is what we're focusing on at the moment. So okay, so that's a lot of the things we've done based on a co-expression analysis. But okay, let me go back to the cell senescence, because what we've done for the cell senescence was also a co-expression network, which is what you see here. So we look at our genes associated with cell senescence, and then we'll build a co-expression network. And as you can see, this network, you know, results in a number of clusters, okay? And each of these clusters is enriched or is particularly associated with different functions: cell division, DNA repair, genes associated with the immune system, etc. Which fits our understanding. So we have a lot of genes associated with cell senescence in these networks, but what we can do is that, well, they cluster in particular groups associated with particular processes and particular functions. So then when we did various analyses, we used, you know, the guilt by association analysis. We actually had gene expression data for cell senescence, which I won't go into detail. We did various network analysis, looking at also at hubs. So these are, you know, genes central in the network, what's called bridging adapters, which are genes that connect different parts, different groups of the networks, and so on. And we derived a number of candidate cell senescence genes. And then in collaboration with Claire Bishop in London, we tested this experimentally in cells by RNAi. I mean, and what you see here are all our candidates and various markers of cell senescence, like well, particularly cell number, cell area, and then markers like P16 and P21. And what is interesting, of the 26 genes tested, 6 of them can be considered strong hits. That is, the manipulation in cells results in strong induction of various markers of senescence and 12 moderate hits. So again, this shows that we can employ these network approaches to identify new regulators of cell senescence. And I mean, as I said, so we published this just a few weeks ago. So Roberto is the first author, but there were actually several other people from the lab involved. This was a project actually started 5 years ago when Robbie Takuto was still in my lab. So he was actually one of the people who started it. Emily was involved. Javier, which was a master student, was involved, and many others were involved in this project. And we did other analysis I don't have time to go into now. So. But as I said, you know, have a look at the paper. There's a lot of results to it. So the other thing I guess we've done quite a lot, and I mentioned before, is DNA sequencing. You know, the costs of DNA sequencing have dropped dramatically in recent years. This is an exponential scale, by the way. So we do a lot of DNA sequencing, genome sequencing that I won't go into detail today, and also RNA sequencing. I guess our first approach was this paper we published already some years ago, but at the time it was the first whole transcriptome sequencing of normal aging in mammals. So we did this in rats, and I still think it's quite an interesting example of applying RNA-seq, because we identified a lot of what called dark matter transcripts, so transcripts that don't map to exons. We identified new genes, so identifying a lot of results that you wouldn't discover with traditional microarrays. So it was quite interesting in that regard. And I mean, we did this with it was mostly the work of Shona Wood, and also collaborating with Brian Murray, who retired a few years back. I mean, now we're also doing RNA-seq specifically in the muscle, in particular to identify microRNAs associated with aging in the muscle. I mean, this is still an ongoing analysis, but there's some candidate microRNAs that seem quite interesting that we want to test in vitro, and this is the work of Ghulam, who is a CIMA-funded PhD student in our lab. So we hope to identify some microRNAs that modulate or that contribute to aging in muscle. As I said, Ghulam aims to test this experimentally in vitro models. But I thought I would point out a paper we just published a few weeks ago on microRNAs modulating Alzheimer's disease. So this was something that started about 6 years ago, I think. So basically what we've done is, so identified some candidate microRNAs important in Alzheimer's disease, or we think are important in Alzheimer's, and then using a mouse model of Alzheimer's disease, the called triple knockout model of Alzheimer's, we injected lentivirus with microRNA into the brain, particularly MER31, and this results in an improvement of the symptoms of Alzheimer's disease in these animals, including improvement in cognitive function and behavior. So we published this just a few weeks ago. This was, so the first author is Ana Viegas, and she was a PhD student with me. As some of you know, I have a secondary affiliation with the University of Coimbra in Portugal. So she was in Liverpool a few years back, but then she did all of these mouse experiments in the Center for Neurosciences in Coimbra. And so she did all of the the Alzheimer's disease model there. But I thought this was quite an interesting work of going from, you know, in vitro work and computational work all the way to testing these particular microRNAs in the brains of mice. So the other thing that in terms of RNA-seq that we're quite interested in now is in terms of single-cell RNA-seq. So, I mean, essentially what you can do is you can isolate individual cells from particular tissues, and you can sequence the individual cells, and then you can map the transcripts and the genes to particular cell lines. So you can have gene expression profiles that are specific to a cell, they're specific for a particular cell type, and then you can compare gene expression not just between conditions, including aging, but also between different cells and between different cell types within a tissue. So that's something that we're quite interested in. We're using data from particularly from something called tabula muricenes and other available data sets. So we're not doing sequencing ourselves, we're using data from others, but trying to identify some patterns in the data, including in terms of cell communication, cell senescence, and so on. And this is mostly the work of Cyril, who is trying to analyze this data. Now, in terms of RNA-seq, the other thing, actually the last thing I will point out is we've also looked into caloric restriction. So we've done this experiment again by Shona and in collaboration with Brian Mary, looking at differential expression in aging and caloric restriction in the brains of rats. We found a lot of pathways you'd expect, like oxidative stress responses, neuroprotection, a lot of epigenetic, chromatin-related genes. And then because Brian had these switches between caloric restriction and ad libitum, we actually had different groups of rats with different lifespans. So we could identify specific genes associated with life extension. So I think we had 110, I don't remember the exact number. I think it's a little over 100 genes that are specifically associated with diet-induced life extension, which I think was quite interesting. We did some microRNA sequencing as well. There was one microRNA, miR98-3P, that was the only one I think was differentially expressed in caloric restriction or in this diet-induced life extension. I mean, we did some follow-up on it. We think there's some epigenetic mechanisms at play, and Shona at the time did some experiments in brain cells to confirm this. So again, I won't go into detail, but I think this, we did various experiments in this, and we published it a few years back. I think it's a good example again of applying applying RNA-seq to gather insights on aging and its manipulation. The other thing I think related that we've been doing is, and this was something we started a few years back, is okay, we have all of these gene expression signatures. We have aging, we have dietary restriction, we have lots of gene expression signatures. So we thought, well, okay, you'll have public databases of gene expression signatures of drugs. So can we identify drugs that have similar gene expression profiles to aging or to dietary restriction? And so we try, as I said, we tried this a few years back. I mean, it's fairly straightforward. So, but it was quite surprising that we found a lot of over 1,000 drugs tested. I mean, the top one was Repurposing, which we actually know extends lifespan in mice. So we went and actually we tested this in C. elegans. This was mostly the work of Sean Calvert, who was a PhD student in our lab. And of the 5 drugs tested in worms, 4 of them extended lifespan. So that was quite interesting. Again, this showing that this what I call this network pharmacology approach can lead to repurposing of compounds for life extension. So I mean, we published this a few years back, but then since then, I mean, I thought the most interesting compound was allantoin. So allantoin is actually a marker of oxidative stress, and it's using anti-aging skin creams already. But we're the first to show it extends lifespan, and we've been focusing on its mechanism since, and also of other similar compounds. And I mean, the person doing this has been Dominic, who's recently finished his PhD, and he generated a lot of results trying to understand the mechanism of allantoin and related drugs And his hypothesis is that they work via autophagy, and he did various experiments that I mean, we're still going through all of his results, and we hope to publish those soon. And I won't go into detail now, but basically we did some mechanistic follow-up on the drugs that we discovered. Now, the other approach in terms of computational that we've been focusing on in recent years as well is machine learning or artificial intelligence. We did this review a few years back about applying machine learning to aging. So if you're interested, it reviews various works and various approaches. I mean, basically, I think machine learning, I think it was Daniel Palmer who used to work in our lab who said that machine learning is a bit like if you go to one of those websites to determine who your Game of Thrones character is, it uses a machine learning approach. So for example, if you say, well, do you like killing people? And if you say yes, then well, you're going to be more related to certain characters of Game of Thrones than others. So machine learning employs those features, only instead of being a view to determine your Game of Thrones characters, it applies the features of genes to determine those that would be related to aging, or well, actually any phenotype really. So it will look at the features of particular genes that have not been studied yet in the context of aging to try to identify those that are more likely to be related to aging, and also to identify which features are associated with aging. As I said, so Daniel, until recently, he was in my lab. Now he's a postdoc in Germany, but he was involved in a lot of our machine learning approaches, and a lot of this is done in collaboration with Alex Freitas. Actually, Alex is was actually a student in my lab over 10 years ago, and is now a professor at the University of Kent. But a lot of it is done in collaboration with him. And just to give one example of how you can apply this, and this is the work with Alex and Daniel. So we can use this to predict new genes associated with cognitive function. So we can look at genes associated with cognitive function and aging, and we can look at the features, and we can try to predict new genes. So we found 10 candidates, which we tested in collaboration with Yandiri, one of which was a potential new hit, i.e. it seems genetic variants, there is an association with cognitive function and aging in this gene, or a potential association. And I mean, we have a preprint on this in bioRxiv, also looking at gene expression patterns with aging in muscle, heart, and brain. And we have another manuscript in preparation focused on these results of machine learning. So I don't have time to go into detail. The only other thing I would say is that we actually have two relatively new PhD students in my lab, Thomas and Gustavo, quite interested in applying machine learning to aging. Thomas is trying to focus on also images, applying machine learning to images, and Gustavo focusing also on dietary restriction and life extending interventions. So we're trying to continue to apply the machine learning and artificial intelligence methods to gather insights on aging. I mean, the other thing we've done in recent years as well is try to incorporate more human data into our analysis. I mean, we developed a database called Longevity Map of genetic studies associated with longevity. We've also done studies of aging phenotypes using human data sets. So Framingham Heart Study data, we've published a study on that already. We have access to UK Biobank. We've been looking at, for example, if patients taking particular drugs that we find in worms, do they have a younger epigenetic clock? So this was something Dominic explored in collaboration with King's College London and Tim Spector in particular in his Twins UK study. So we want to, I'm interested in applying what we know about aging to human beings. I mean, I think the ultimate goal is really to intervene in aging. I've always said that I didn't become a scientist just to get grants and papers. I became a scientist because I want to discover something. So what really I would like is, okay, we have this normal human life score, but we would like to extend it. We would like to expand the healthy years of life so people extend human health span. And in this context, I just briefly point out a paper we did in Science last year together with a number of collaborators. This was actually done by Stuart, who's has an affiliation with our lab. So the idea is that we classify senescence with the World Health Organization as a way to facilitate trials for human aging and for longevity. Is that we have a classification of senescence. So that's something we know. Obviously, the World Health Organization have other priorities right now, so it's not something that's going to happen right away. But we're hoping in the next couple of years, this is something we can gradually do. So, for example, we're trying to focus on immunosenescence at the moment, given the COVID-19 situation. Can we get a classification of immunosenescence with the World Health Organization? And I think that would be a good first step. So, so in summary, I've told you that, okay, we know a lot about aging manipulations and model organisms. There's over 2,000 genes in our GeneAge databases, and hopefully I persuaded that biology is becoming a mathematical problem. We do need the analytical tools. We need to do the bioinformatics, the statistics, the computational biology, the systems biology approaches. I hope I persuaded you that network analysis can lead to new insights, including new processes, new genes, new drugs. And particularly I focus on our analysis of cell senescence and gene networks, which we've recently published and revealed a number of insights. I've told you about RNA-seq in various contexts. I hope I convinced you that it has advantages compared to microarrays. Although I would say, I forgot to say that, RNA-seq does have a disadvantage, which is the data analysis is more complicated than microarrays. I think microarray is very simple. RNA-seq is a little more complicated. I mean, we have a pipeline to do it. I mean, if everyone is interested in doing any RNA-seq and has any questions, feel free to let me know. I told you about our growing application of RNA-seq methods, machine learning methods. And lastly, I told you about our human data integration, which is something I see we're doing more and more. We want to collaborate with clinicians. We want to employ human data. We want to go from basic science in model systems and model organisms to the clinic. So with that, I think I mentioned everyone's work in our lab. Of course, I think we've been very productive in the past few years, and this is obviously thanks to a number of very talented and ambitious students and postdocs. And of course, thanks to funding, in particular, the Wellcome Trust have been wonderful to us. Also, Liverhome, BBSRC, Royal Society, and CIMA, Allison Foundation, Methuselah, a number of funding bodies that have helped us. And if you have any questions, please let me know. Thank you.
Thank you very much. It's very interesting. There are a number of questions in the chat box, and we can go down them. So George asks, what is more effective, increasing a gene that increases longevity or deleting a gene that decreases longevity?
That's a good question. I think, so I would have to go with decreasing a gene that decreases longevity. So if you look at all of the main life extensions you see in animal models, they are knockouts. Having said that, there is a bias because it's easier to do a knockout than it is to do an overexpression. So there are more experiments with knockouts than there are experiments with overexpression in our databases, and I mean I would assume in PubMed. So I think there are some biases, but at the moment, if you look at the top life-extending interventions, they are knockouts. So it is disrupting a gene that is anti-longevity, like growth hormone, IGF-1 pathway.
But if you did that, do you not then, going back to your cancer question, increase the risk of cancer?
So you're saying like can a gene associated with longevity also increase the risk of cancer?
Yeah.
So for the growth hormone pathway, you see, I mean, the knockout mice for growth hormone, they tend to be cancer protected. So they tend to be quite small. So there is a question of whether, is their life extension mostly due to cancer protection? I've asked that question to people like Richard Miller and David Harrison and Arlen Richardson, and they said, no, there's other aspects. You are retarding aging. Yes, they're cancer protected, but there's other aspects of aging that are retarded. So answering your question, yes, when you manipulate aging in mice, you also affect cancer, but that's not the whole story, I would say.
So I'm not sure, Connor, was your question answered further down in the chat? Connor, do you want to come online? We'll go to Kevin's question then. So Kevin says, with respect to gene friends, does the rate at which the assumption that co-regulation of transcript abundance equates to functional outcome reflect the evolutionary age of the gene?
Does the rate? I can see the question.
Okay.
Co-regulation turns out equates to functional. So that's a good question. Does the how? So. You know, I think we have looked into that actually. So I think Gianni looked into that. So we published a paper in BMC Evolutionary Biology in 2015 where we looked into that. I don't remember the answer, however. My recollection of that paper was that, so what we did in the paper, we compared co-expression networks between mice and humans. That's what we did. And my recollection was that there were some genes that were actually well conserved, that they had quite distinct co-expression patterns. But I don't remember the overall message. I would assume that better conserved genes, i.e. genes that have older genes, let's say, would be more co-expressed, or their co-expression would also be more conserved. I would assume, but I'm actually not 100% sure.
So Priyanka's added the reference further down in the chat, so
okay, that's great.
People can link to that.
Yes, I think checking that paper would be the best. It's been a few years. I don't remember exactly what was the answer, but I think the answer will be there. Yes.
So Nick asks whether metformin was one of the 5 drugs you identified as pro-longevity.
No, metformin was not one of them. It was not in the top 20, I believe. So we haven't tested metformin. I think other people have tested metformin in worms, but we haven't. No.
So it's been looked at as retarding, as a retarding treatment for AMD. So it might be interesting.
Yes, I think, well, there's a lot of interest in metformin in the field at the moment. There's an attempt, clinical trial near Barzilay, the first big clinical trial for aging to focus on metformin. So there's a lot of interest, but we haven't looked at metformin. I mean, the other thing we haven't done, I should say, is look at drug combinations. So, does allantoin and rapamycin extend lifespan more than together? They extend more than each of them individually. Actually, my colleague Jan Gruber in Singapore have tested this, and the answer is yes. But that's okay. So that's one particular combination. But it would be very interesting in looking at drug combinations, actually, including with metformin.
Any more questions? Kevin, did you want to follow up on that question? Oh, there's another question. Oh, Kevin says no thanks. Brendan says, I may have missed it, but what was the direction of association between allantoin and longevity? Was this a pathway level association?
So the short answer is we don't know for sure. I mean, so we initially identified allantoin because the gene expression patterns induced by allantoin in a cell line, in a human cell line, were similar, overlapped statistically significantly with the gene expression patterns that you get with caloric restriction. And actually, I mean, that was caloric restriction in a cell model. You actually take. So this was done by, I think, what's his name? It's a Spanish guy in the NIA, Rafa de Cabo. He actually took blood from monkeys under caloric restriction, and he took serum from them and he put it in cells, and that he looked at the gene expression. So that's what we see. We found allantoin because the gene expression patterns of were overlapping significantly between allantoin and dietary restriction. Exactly how allantoin works, we don't know. So allantoin definitely extends lifespan. So, if you feed allantoin to C. elegans, it extends lifespan. I mean, Jan Gruber in Singapore has shown that as well. But exactly which pathway is involved, we don't know. Dominic has done some experiments as part of his PhD, suggesting that, well, his hypothesis is that autophagy is involved. And, you know, he's done a number of experiments on that. So that's I would say, our stronger hypothesis is that the pathway involved of the mechanism would be autophagy, but we don't know for sure.
Okay, well, I think we'll probably have to stop it there because people will have to go to other meetings. But thank you very much, Pedro.
Thank you.
I think that's been obviously generated a lot of discussion, which is always good.
Well, thank you very much. And I'd just like to say I'm glad Roberto likes my shirt. You know, that's, I mean, that makes my day.
Makes your day. Good, good. And hopefully see everybody again next week. Thank you very much, everybody.
All right. Thank you. Bye. Stay safe.